Hello and a very good day, everyone. I am Nursaffa Alisya from University Putra Malaysia. Will be presenting my research project on the title of a new pharmacology-based investigation on the bioactive compounds and molecular mechanism of Moringa oleifera against pancreatic cancer. Pancreatic cancer is usually associated with a very poor prognosis. The 5-year relative survival rate of pancreatic cancer has shown a slight improvement around 10% from 2014 to 2021. However, the number of deaths in pancreatic cancer is still increasing since the majority of the patients are diagnosed at the advanced stage. Moringa oleifera, which mostly found in India, Africa, and Southeast Asia, has been used in traditional medical systems such as Ayurveda and traditional Chinese medicine to treat various kinds of diseases. Interestingly, the in vitro study of Moringa has revealed 98% reduction of pancreatic cancer cell survival. This present study aims to evaluate the potential effect of Moringa against pancreatic cancer through neuropharmacology. For methodology, the study was divided into 3 steps, which are target identification, screening potential target, and neuroanalysis. In the first step of target identification, the identification of Moringa compound were performed by using PCM-SP and PCM-ID databases. However, its constituent cannot be found in the database. Alternatively, studies related to Moringa were searched through PubMed to determine the compound, and the compounds will be taken from Pong et al. 2020 study. As the compound will be orally administered, they need to undergo the IDME screening to predict the oral bioavailability, drug likeness, and Kakoto permeability. The bioactive compounds that meet the requirement will then be predicted the target genes using drug bank, PGIDP, and CTG databases. Initial screening of the target genes were also conducted for the remaining compounds that cannot be found in the TSM-SP, and the bioavailability was searched through PubMed. For pancreatic cancer, OMIM and Malakat databases will be used to collect the target genes. The result for the target genes will then be presented by the Venn diagram to determine the pancreatic cancer-related target genes of Moringa oleifera. In the final step, the pancreatic cancer-related target genes of Moringa will be imported into string database to study the protein-protein interaction. Then, the interaction of the protein will be analyzed by determining the gene ontology and pathway analysis using David database and constructing compound target pathway network using Cytoscape. As a result, there are 32 bioactive compounds in Moringa that were listed in one study. However, only 4 compounds, which are catechin, epicatechin, quercetin, and KF were selected as they met the requirement of the drug screening. For the screening of the remaining Moringa compounds, only 3 out of the 25 compounds were found. Glucomoringin and glucoraphanin were reported to have a good bioavailability, while the solubility of moringinin can be improved by the formation of salts. Next is the figure that shows the result of pancreatic cancer-related target genes of Moringa oleifera and its protein-protein interaction network. There are 81 nodes that overlap between Moringa and pancreatic cancer, and 373 edges where the nodes represent the target genes, while the edges represent the interaction between them. This network showed that these target genes have complex interaction between each other. From the PPI network, 9 hub genes were identified based on degree, closeness, and betweenness value, which are listed in Table 3, including TP53, AKT1, VEGFA, MAPK3, IL6, JUN, MAPK1, CCND1, and TNF. Hub gene is defined as node with high connectivity, which is considered to play a crucial role within the network. Next is the result of gene ontology and pathway analysis. The result demonstrated that the most enriched biological process was associated with the regulation of apoptotic process. It has been reported that positive and negative regulation of apoptotic process are simultaneously activated through Norsyasya and protective signal via various pathway. Therefore, it is pertinent to finally balance the regulation of the 2 signals to increase the effectiveness of Moringa against pancreatic cancer. In the CAP pathway, the enrichment pathway were mainly associated with cancer-related disease, including pancreatic cancer. Last but not least is compound target pathway network, which is constructed to show the interaction between the compound, pathway, and the target genes. This network contains 98 nodes, which comprise of 7 compounds, 81 target genes, and 10 pathway with 1,623 edges. From the compound target pathway network, 2 hub compounds, which are catechin and KF with the average node degree more than 30.909, were identified as potent compounds responsible for the therapeutic effect of Moringa against pancreatic cancer. As for the discussion, 2 bioactive compounds, catechin and KF act in a wide range of pharmacological properties, including anti-cancer. This compound play a role in the regulation of various cancer cell activities by inducing the apoptosis effect. The 9 hub genes identified in the study also play a crucial role within the network with high biological importance and significantly enriched in multiple pathways. Previously, studies have shown the mutant TP53 induced gemcitabine resistance towards pancreatic cancer cells. Therefore, Moringa could be the new therapy in treating pancreatic cancer since it is targeting TP53, the common mutated tumor suppressor gene in pancreatic cancer. In conclusion, this in silico study has revealed the multi-compound, multi-target, and multi-pathway and provide a theoretical insight on Moringa intervention in pancreatic cancer, which suggests direction for further experimental validation and subsequent clinical application to verify this finding. These are the reference. With this, I would like to thank all of you for giving your attention and listening to my presentation. Thank you.